Okay, so welcome to this next video in which we are discussing the phospholipase C beta enzymes. Okay, so sorry for the abrupt ending to the previous video. We were discussing alpha subunits of heterotrimeric G proteins and the fact that they all get palmitoylated. They get palmitic acid molecules stuck onto the side of them, and these palmitic acid molecules are responsible for their insertion into the underside of the phospholipid bilayer. Okay, and they hold the alpha subunit dangling underneath the underside of the phospholipid bilayer. Okay, right. So all alpha subunits get a palmitic acid molecule attached to them. However, some alpha subunits not only get a single palmitic acid molecule attached onto them, but they also get another lipid moiety attached to them known as a myristic acid molecule. Okay, so once again, what's the new name for myristic acid? Myristic acid's new name is tetradecanoic acid. Okay, and once again, this is a much better name because it tells us exactly what we're working with here. It tells us we're dealing with a 14-carbon fully saturated carboxylic acid. So here's our carboxylic acid group once again, and this time we're going to have 12 methylene groups. So I'll draw one methylene group and then bracket it and subscript it 12. And then on the end of the 13th carbon then, you'll then have a methyl group coming off the end. So this is myristic acid or tetradecanoic acid. Certain alpha subunits also get myristic acid molecules attached onto them, okay, as well as the palmitic acid molecules. So they get two lipid moieties stuck off the side, which both implant into the inner leaflet of the phospholipid bilayer and then therefore involved in holding this alpha subunit to the underside of the phospholipid bilayer. Okay, right. So the alpha subunits that get myristic acid molecules attached to them are the alpha subunits within the G alpha I/0 family of alpha subunits, and we'll come to what that family is in a moment. Okay, right. So now we've discussed the alpha subunits of heterotrimeric G proteins. Let's move on to the two other subunits of heterotrimeric G proteins. So the two remaining subunits of heterotrimeric G proteins are called the beta and the gamma subunit, and these never come apart under physiological circumstances. Okay, so they are often collectively referred to as the beta-gamma complex because they're always bound to one another. Okay, now, when the alpha subunit is in the off state but not in the on state, okay, the alpha subunit can bind to a beta-gamma complex. So since our alpha subunit is in the off state and has guanosine diphosphate bound to it, it can associate with a beta-gamma complex. Okay, so here is our beta subunit, here is our gamma subunit, and they're bound to one another. So we'll have the beta subunit in blue here, okay, and then we'll have the gamma subunit in green. Okay, now, the gamma subunit also has a lipid modification attached to it. Okay, so the gamma subunit has a lipid moiety attached onto one of its amino acids, and it's exactly the same principle as for the alpha subunit. This lipid moiety is going to have an extremely hydrophobic tail, which will then implant into the inner leaflet of the phospholipid bilayer and therefore hold the beta-gamma complex dangling on the underside of the phospholipid bilayer like so. Okay, now this is a slightly more complicated lipid moiety than palmitic acid and myristic acid. This is what's known as a geranyl group. But the principle is exactly the same, that it's a hydrophobic group that is implanting into the inner leaflet of the phospholipid bilayer and thereby holding the gamma subunit, which indirectly then holds the beta subunit up, attached to the inner leaflet of the phospholipid bilayer. Okay, right. So, here then is our heterotrimeric G protein. What we now want to discuss is the diversity of heterotrimeric G proteins. We want to discuss the fact that there are multiple different alpha subunits, multiple beta subunits, and multiple gamma subunits. Okay, and therefore the scope, to get some sort of appreciation of the scope of the different heterotrimeric G proteins. Okay, we also want to see the forms of heterotrimeric G proteins which are actually going to activate our phospholipase C beta enzymes. Okay, then what we'll do is we'll discuss the G protein cycle in full generality, and then we'll specialize back down to this G alpha Q/11 alpha subunit family and see how it can activate our phospholipase C beta enzymes. Okay, right, so let me get another piece of paper. So, we want to discuss how many different heterotrimeric G proteins there are. So we're going to begin with the alpha subunits of heterotrimeric G proteins, okay, more correctly called the G alpha subunits. Okay, so there are 16 different genes for alpha subunits, okay, and these code for a total of 21 different known alpha subunits. Okay, so 16 genes which give us 21 different subunits. So at first you might think that this looks like a bit of an anomaly. How can you have 16 genes giving 21 subunits? But we've seen exactly this already. We've seen those phospholipase C beta enzyme genes give multiple different proteins. So this is the phenomenon of splice variants again. So some genes will have multiple splice variants and therefore give rise to a greater number of alpha subunits than just one. Okay, so we're going to go through the 16 different genes of alpha subunits. We're not going to dwell hugely on the different splice variants. Okay, right, so the alpha subunits of heterotrimeric G proteins then, they are grouped into 4 great families, okay, to help us understand them. So the 4 families of alpha subunits are known as the G alpha S family of alpha subunits. Then we have the G alpha I/0 family of alpha subunits, which are the ones which get myristoylated. Okay, then we have the G alpha Q/11 family of alpha subunits, and then finally the G alpha 12/13 family of alpha subunits. So these are the 4 great families of alpha subunits. Okay, right, let's start off with the simplest family, the G alpha S family. Okay, this contains 2 members. It contains the G alpha S gene and also the G alpha ULF gene. Okay, now G alpha S has a huge number of different splice variants which we're not going to go into. Okay, G alpha ULF just has 1 splice variant. Now the reason these 2 are grouped into this first family together like so is because they both have very similar functions. Okay, all of the splice variants of G alpha S and the 1 splice variant of G alpha ULF, when they are activated, okay, so they're alpha subunits, so they have these 2 states, the off state and the on state. When they're in the on state and have guanosine triphosphate bound to them, these alpha subunits go off and activate adenylyl cyclase enzymes. Okay, so G alpha ULF has a very similar function to G alpha S. G alpha S is more famous, and the reason is that you find G alpha S all over the place in the body, whereas G alpha ULF you find in very restricted places within the body. For instance, the tissue of the olfactory epithelium, hence its name G alpha ULF rather. Okay, and in these special tissues which have G alpha ULF, such as the olfactory epithelium, you find very low levels of G alpha S. So basically in these tissues, what happens is G alpha ULF effectively replaces G alpha S. So instead of having G alpha S, you have G alpha ULF, but effectively it does exactly the same thing. Right, so those are the only 2 members within this first family. Now down to the second family. The second family is the most difficult family. This one has 8 different members. Okay, so firstly there are the G alpha I's. Now there is not just 1 G alpha I. There are 3 separate genes for alpha I subunits. Okay, there is G alpha I1, there is G alpha I2, and there is G alpha I3. Okay, when someone says G alpha I, what they mean is 1 of the 3. Okay, so all 3 of these pretty much do the same thing. They go off and inhibit certain adenylyl cyclase enzymes. Okay, so often people don't distinguish between the 3 different genes for G alpha I subunits, okay, and instead they will just refer to G alpha I, and they'll mean 1 of the 3. okay, because they all pretty much do the same thing, and often a G protein coupled receptor which activates one will also activate the others as well. Okay, so it doesn't make too much sense to distinguish between the three, but they are three separate genes. Okay, right. Next up, we have very related alpha subunits. So we have G-alpha O, okay, which again has a very similar function to these three G-alpha I's. It too can inhibit adenylyl cyclase enzymes, okay, and it's the other one after which the entire family is named. Then there's also G-alpha Z, which again has similar functions to the three G-alpha I's and now the G-alpha O, in that they inhibit adenylyl cyclase enzymes. Okay, right, so those five are all very similar. Next up we have two that are very different, okay, but they're still grouped into the same family for reasons that we'll see. Okay, the next two are G-alpha T1 and then also G-alpha T2. Okay, so what do these two do? Well, basically, in the photoreceptive cells of the retina, there are two main photoreceptive cells in the retina, rod cells and cone cells. Okay, and in these rod and cone cells, you have receptors for light. Okay, in rod cells you have rhodopsin, in cone cells you have iodopsin. Okay, and these receptors are G protein coupled receptors, and when the light activates these G protein coupled receptors, they are going to interact with a heterotrimeric G protein. And the heterotrimeric G protein which they interact with is known as a transducing G protein. Okay, so the alpha subunits of the transducing proteins are these two here. Now in rod cells you have a different type of transducin between cone cells, and the difference is in the alpha subunit. So in rod cells you have the G-alpha T1 subunit as your alpha subunit of your transducin protein. So G-alpha T1 is the alpha subunit of rod transducin. Meanwhile, in cone cells, you have the G-alpha T2 alpha subunit as your alpha subunit of your transducin protein. So in cone transducin, G-alpha T2 is the alpha subunit. Okay, so these collectively are called the G-alpha T's. Okay, so collectively they are called the G-alpha T's. Now, why are the two G-alpha T's grouped into this family with these five alpha subunits that inhibit adenylyl cyclase enzymes? Well, the G-alpha T's do not inhibit adenylyl cyclase enzymes, but what they do is they activate phosphodiesterase enzymes. And specifically, they activate phosphodiesterase enzymes which cause the breakdown of cyclic GMP. Okay, so let's draw the parallels here. These five inhibit adenylyl cyclases and therefore make cyclic AMP go down within the cell by stopping its production. These two make cyclic GMP go down in the cell, so not cyclic AMP but cyclic GMP, by activating an enzyme which destroys the cyclic GMP. So that's the relation between them. They both make cyclic nucleotides go down, so that's why these two G-alpha T's are grouped into a family with these five alpha subunits that inhibit adenylyl cyclase enzymes. Okay, so that's 7 now. The final one, which I'll just squeeze in here, is known as G-alpha GUST, and this is involved in the transduction of gustatory stimuli. Okay, so it's involved in the taste system. Okay, so those are the 8 alpha subunits that are all grouped into this G-alpha I/0 family of alpha subunits of heterotrimeric G proteins. Next up, we have the G-alpha Q/11 family, which is the one we're really interested in. Okay, so this is a big family for us. The others I'm just doing for a complete discussion. This is the family that's going to actually be involved in activating phospholipase C enzymes of the beta form. Okay, so you have 4 members in this family. The most famous member by far is G-alpha Q. Okay, but you also have G-alpha 11, the other one after which the entire family is named, then G-alpha 14, and then finally G-alpha 15/16. Now, all of these 4 alpha subunits are involved in the activation of phospholipase C enzymes of the beta form. Okay, it is not just a job of G-alpha Q. It's not even just a job of G-alpha Q and G-alpha 11. All 4 of these, G-alpha Q, G-alpha 11, G-alpha 14, and G-alpha 15/16, when in the on state, can activate phospholipase C enzymes of the beta form. Okay. Now, I would just like to tell you the story behind how G-alpha 15/16 became known as G-alpha 15/16, because I don't want you to go away thinking, is this 2 alpha subunits or what? Why is it called that? Well, basically, there were 2 groups of researchers. One was working in mice, and they discovered a new alpha subunit of the heterotrimeric G protein, and they called it G-alpha 15. Okay, so this was the alpha subunit in mice. Then another group of researchers was working in humans, and they discovered a new alpha subunit of heterotrimeric G proteins, and they called it G-alpha 16. Okay, later it transpired that G-alpha 15 and G-alpha 16 are the same thing. This is the mouse equivalent of the human G-alpha 16, and this is the human equivalent of the mouse G-alpha 15. Okay, so there was then a movement to have the whole thing renamed G-alpha 15/16 in both mice and humans, because we don't want it called something different in mice from humans. So the movement was then to rename both of them G-alpha 15/16. Now, it hasn't caught on as well as you would hope. People working in mice still refer to it as G-alpha 15, and people working in humans still refer to it as G-alpha 16. But G-alpha 15/16 just embodies both of those names. Okay, right. So that's the family of alpha subunits that are capable of activating phospholipase C enzymes of the beta form. Okay, now G-alpha 12/13 then is the family that no one has ever heard of. Okay, this contains 2 members, G-alpha 12 and G-alpha 13. Okay, and these 2 alpha subunits are involved in cytoskeletal remodeling. Okay, and that's all I'm going to say about those. Right, so let's now check that I have told you about 16 different genes of alpha subunits. So we have 1, 2, 8 in this second family, that takes up to 10, 4 in this third family, that's 14, 2 in this final family, that's 16. As I say, we're not going to dwell on the different splice variants, because the different splice variants pretty much do the same thing, so it's not too important. Okay, so those are the 16 different genes of alpha subunits, and the ones that we're going to be particularly interested in are these 4 here. Okay, right. Now, just for a completion of the discussion of diversity of heterotrimeric G proteins, I'm also going to tell you about the different G beta subunits and the different G gamma subunits. Now, don't worry, this won't take us long. Okay, so G beta subunits next. There are 5 different genes for G beta subunits, and these in total make 6 different G beta subunits. Okay, so we know exactly what has happened here. One of these genes has got 2 splice variants, and that is exactly what has happened. Okay, so the 5 genes then are named G beta 1, G beta 2, G beta 3, G beta 4, and G beta 5. Okay, now which of those 5 genes has 2 splice variants? Well, it's G beta 5 that has 2 splice variants, and the 2 different splice variants of G beta 5 are called G beta 5/5, okay, and then also G beta 5L, okay, right. Now, that's the only gene that has 2 splice variants. The other 4, as far as we know, only have a single splice variant. So G beta 1 produces the protein G beta 1, G beta 2 gene produces the protein G beta 2, etc. Okay, it's just with G beta 5 that you have to be careful about the naming of the actual proteins, because there are these 2 splice variants here, G beta 5/5 and G beta 5L. Okay, right. Now finally, let's go on to the G gamma subunits. Okay, so the G gamma subunits are nice because splice variants aren't a big thing for them. Okay, so there are 12 genes for gamma subunits, and there are only 12 known subunits. Okay, so each gene codes for a single gamma subunit. Okay, now the naming is almost perfect with one little blip in it. Okay, so there is G gamma 1, then there is G gamma 2, G gamma 3, G gamma 4, all the way up to G gamma 5. There is then no G gamma 6, which is a shame. You then go straight on to G gamma 7, and then it goes on all the way up to G gamma 13. So you have G gamma 8, G gamma 9, G gamma 10, G gamma 11, G gamma 12, and then finally all the way up to G gamma 13. So you have to go beyond 12 because we've missed one out down here. Okay, and those are the 12 different genes for gamma subunits, but they all code for a single subunit, and they code for a subunit that is given the exact same name. Okay, right. So now what I'd like to say is a little thing about the naming of heterotrimeric G proteins. So what you might think would be ideal is if, when naming a heterotrimeric G protein, people named up exactly which alpha subunit they were dealing with, exactly which beta subunit they were dealing with, and exactly which gamma subunit they were dealing with. An example would be G alpha I1, beta 1, gamma 1. Okay, which tells us exactly which alpha, exactly which beta, and exactly which gamma subunit are in this heterotrimeric G protein. People rarely do this. Firstly, because they rarely know, and secondly, because they don't need to know, they don't care that they don't know. The reason is that it doesn't, well, as far as we know, it doesn't hugely affect the signaling capability of the heterotrimeric G protein, which beta and which gamma subunit you have. Now, this is probably ridiculously naive, in fact, it's almost certainly ridiculously naive. But at the moment, our current position is that which beta and which gamma subunit you have doesn't hugely affect your signaling. Okay, so people don't really care which beta and which gamma subunit they have, and they often just refer to it as the beta-gamma complex. Okay, so they certainly don't usually specify which beta and which gamma subunit they're dealing with. Instead, people label their heterotrimeric G protein according to which alpha subunit it has. Okay, so you might see this referred to as a GI1 heterotrimeric G protein, which tells you that the alpha subunit is alpha I1, basically. Now, even that's wishful thinking, because again, people often don't know that sort of information, and also it doesn't matter that they don't know, because usually when you're dealing with these signaling cascades, it's very rare that you have a G protein coupled receptor that will literally only activate heterotrimeric G proteins which have a G alpha I1 subunit as their alpha subunit. Often they will activate a much broader repertoire of G proteins. Okay, so often people will just call this GI to say that the alpha subunit is one of the I's, basically. Okay, and the reason that that makes sense is because often G protein coupled receptors will activate all three of the alpha I subunits rather than just one, so it doesn't really usually make sense to talk about a specific alpha subunit. Okay, in addition, people sometimes go even more broad. They label it just by the family that the alpha subunit is in. So they might put GI/0 here to denote that the alpha subunit is within the G alpha I/0 family. Now, if people put something like that, you can assume that really it means one of these five on the top here, i.e. a G alpha I, one of these three, or a G alpha O or a G alpha Z. You can assume it does not mean one of these transducing alpha subunits. If people mean a transducing alpha subunit, they'll call it GT. Okay, one more thing which is particularly relevant for us. Okay, these heterotrimeric G proteins which have alpha subunits within this G alpha Q/11 family, we will denote these as GQ/11 heterotrimeric G proteins. Now, what this means is a heterotrimeric G protein where the alpha subunit is within this family, the G alpha Q/11 family. It doesn't just mean it's G alpha Q or G alpha 11. It means it's G alpha Q, G alpha 11, G alpha 14, or G alpha 15/16. One of those four. Okay, and we'll be using that notation a lot. Okay, right. So we'll have a break here, and then in the next video what we'll talk about is the G protein cycle in general. Okay, so how heterotrimeric G proteins are activated by G protein coupled receptors. And then what we'll talk about is how, once you've got an activated GQ/11 heterotrimeric G protein, that alpha subunit will go off and activate these phospholipase C enzymes of the beta family.